A little bit of background. Bachelor of Science in Biology with a research thesis in engineering science minor. So it started out going crossing disciplines. University of California, Berkeley, cell developmental biology and protein chemistry. Now here since August, Department of Biology with Forensic and National Security Sciences Institute. The research focus in my lab has been for roughly the last 35 years is how does a cell make a decision? How does a cell decide to divide? Or decide that this is danger and I need to neutralize it? Our research support is quite broad, from the National Science Foundation, National Institutes of Health, and the Defense Drug Production Agency, part of the Department of Defense. I served as advisory consultant to a number of agencies, and currently have 2 issued U.S. patents, 1 pending, and 6 that have been submitted to the university for consideration. So we're here at Syracuse University, and we are really fortunate to be here. Now the motto of Syracuse University is Sus Cultores Scientia Claravant. We all know that, right? We all learned the Latin. Knowledge crowns those who seek her. Even Otto knows that. In the College of Arts and Sciences, and in my teaching, one of the things I like to do is understand the power of words. I'm not an English major, I'm teaching languages. But let's look at the words that we've just gone through. University, if we go back to the Latin roots, means guild. College was to gather. Arts, science, learning, and scholarship. I thought it was just painting and fashion design. Maybe sculpture. Science is to know. So we can take this phrase in College of Arts and Sciences and see that we are gathering to do our science, learning, and scholarship so that we can know and ultimately understand. And at a university, we're all here, it's a guild of these faculty, a guild of the students. We're interested in how cells make decisions. Very simply, how do you go from interphase, or single cell, to 2 cells? If we could start that movie, please. What we're going to see is a digitized version of a movie that was made, I would say, roughly 45 years ago by a colleague of mine on 16mm film. This is how we saw cell division way back then. Not with all the fancy digital computer activated stuff of things, the static images that you see in textbooks, but rather the actual movements of cell division. This is going to be in sea urchin embryos. This is about 30 times real time. We have the nuclei here, which are moving around. We don't understand how that happens. The same thing is happening in dividing cells in your body. In your body. All of this, we have to understand all of this in order to understand how embryos develop into us, how we make blood cells, how we go ahead and interfere successfully with a tumor cell without nailing everything else. Well, it's kind of an interesting challenge, isn't it? We've been at it now since 1870s when it was first described in animal cells. We're just scratching the surface. And this cell now is moving chromosomes, and it ultimately will divide. And it goes on. All that dynamics, we'll stop that, please. So a number of tools that we use, in this case, we work with 2-cell embryos. And we do microinjection. We're injecting a millionth of a liter of solution into a single cell to find out if this compound works in the test tube, will it work in the next cell, and stop cell division, or tell us about the mechanism. About 100 years ago, my academic great-grandfather suggested that calcium somehow was playing a role in controlling cell division. He didn't know how, but everything pointed him in that direction. Good intuition. How to see it? Well, a friend of mine was working with a jellyfish protein that when it binds a calcium ion, 1 calcium ion, it gives off a photon, 1 photon. We just had to figure out how to be able to see single photons coming out of the cell into which we put the jellyfish protein. And asked what was the space-time pattern. And did it correspond to what the cell was actually doing? The solution was this microscope. This is the technical drawings that I did for it. And where we're able to take the photons of 1 channel, 1 band of color, and the structure of the cell in a different manner, and we can see them simultaneously. And I was awarded 2 US patents for that. This is what the cells look like. Here's the white dots are the record of single photons. Now the camera was made, it was a product, but it wasn't seeing single photons. It was seeing packs of photons. And I sat down with the engineer, the company gave us the time, and we figured out how to correct the camera so that it actually saw single photons. Also spent time working with supercomputer specialists who were designing and building supercomputers, and we were doing real-time tera-op computing. Million operations per second. 25 years ago. And this is just a record of that, where you see the individual dots coming up, but they're not, it's not solid, it's in a modeled pattern. And that's very important because it told us something about the mechanism involved. The mechanism pointed us to the chemistry. If we understood the chemistry, we could perhaps go ahead and figure out how the whole thing worked, and in fact, we've done that now. Where we know all these blue boxes are enzymes, and we find not only for cell division, but also how the frontline defenders in the immune system stand up to go ahead and nail that danger that they have to do. And they're using the same system. We have a numerical model that we work with, and lo and behold, much to my surprise and worry, is that the model predicts exactly what we observe. It's all based on data. A while back, an otolaryngologist, a friend of mine, knocked on the door and he said, "Could we look at the motion sensors of the inner ear in a living fish?" I said, "Sure." "You're going to open up the fish for me?" He said, "Well, yeah, kind of." I said, "You're going to take everything out?" He said, "No." I said, "Okay. Here's the problem. Look at the person next to you. Look at the diameter of the hair on their head. The motion sensors of the inner ear, their length is the diameter of a hair on their head. Their diameter is 1,000th of that distance. If this was a wire 1/4 inch in diameter, it would be 38 feet long. This is embedded in tissue that's 1.2 millimeters thick in your skull, in the densest part of your skull. How are we going to see this? Well, here's the problem. Here's the fish skull. There are the motion sensors in here, this little cup. All of the brain is down here. The volume I had to work with to get all my optics, my fancy schmancy optics, was 2 millimeters on the side. What do? Well, you talk to engineers, and you go out and you design and build it. This is what they look like. Let's go back. This is what they look like in the scanning electron microscope. There are the motion sensors, and here they are in video. We actually succeeded. Textbooks say you can't do this. We did it with this microscope. Microscope we designed and built. The tools that I needed did not exist, so I simply went out and created them. Was it easy? No. But patience, perseverance, and a lot of hard work paid off. Isn't that what this entrepreneur business is all about? You have a passion for it? As you can tell, I don't have much passion for what I do. Now, here, I'm working with fashion design students on senior projects. I was working with one this afternoon before coming over here, and I'll be back at the warehouse tonight, and I'll be at the warehouse Saturday and Sunday, so they can be ready for their show on the 25th at the Shine Center. Please all come. We're reaching out of the box, out of a comfort zone, to help dedicated students in college merge fashion, aesthetics, and science. It's going across colleges and disciplines